Hello, and thanks for joining us today. My name is Chrissy, and I'm the marketing manager for Mosaic. So I'll be your host for this webinar. In today's session, you'll hear from our CSO, Eric Furfine, about a protein engineering strategy developed right here at Mosaic Biosciences as a modular alternative for long-acting ocular therapeutics. Eric brings extensive leadership experience across biotech and pharma, including senior roles at 11 Biotherapeutics, at Nexus, and Regeneron, where his work contributed to clinical programs and transformative medicines such as EYLEA. Before we begin, a few quick housekeeping items. The chat will be closed today during the session, but you may submit questions throughout the webinar through the Q&A feature, and we'll address as many questions as time allows. All right, and with that, I'm going to turn it over to Eric.
Well, thank you very much. Glad to have the opportunity to share our pharmacokinetic enhancement program with the audience. So give me one sec. Okay, so here's what I'm going to talk about today. I'll start with a little bit of an introduction into why and how Mosaic came about and what we specialize in. Then I'll move into a little bit of table setting on how we see the landscape of ocular therapeutics, what needs improvements, what kinds of things are going on to improve the ocular therapeutics. And then finally, I'll move the majority of the talk, which is to explain how Mosaic is approaching enhancing ocular therapeutics and developing technologies to improve them. Okay, so Mosaic. Basically, when you want to contract out work at CROs to do part or maybe all of your company virtually, there are a lot of challenges involved, and that the challenges are really based mostly in finding the places to do the work that you want to do and getting them to communicate with each other. And the management of this process is challenging and difficult. Most of these companies don't talk to each other. There's, information that's not being transferred at each step when things are passed from one company to another. And all this together makes it more and more difficult to ensure you get a good outcome at the end of the day and that you find the medicine you're trying to make. And so when I was helping companies do these sorts of things, I was wishing for a place that you could go that didn't require you to go to a bunch of different places, but you could get everything done in one. And so that's what we created at Mosaic Biosciences. We created an end-to-end protein engineering and drug discovery, more generally, organization that allows you to come to us with nothing more than an idea, actually, and get delivered at the end a clinical candidate that's ready to begin IND-enabling studies. And you can start anywhere in this process of making a medicine, all the way back as far as validating the target, figuring out how whatever your science is works, all the way through to engineering the molecule and testing it in vivo and seeing if it does the kinds of things you want to do. We provide the strategic advice and the talented scientists to work in our state-of-the-art labs to get all this to go. We're happy to work on any part of this for you, but maybe the biggest differentiator we have is to be able to provide that end-to-end service. So what are the things that we do in Mosaic? There's a lot of protein engineering design. That's probably the thing that we do most for our partners is protein engineering work, making protein therapeutics. We have an antibody discovery platform that has two parts. It has an in vitro part where we can do selections using phage and yeast display. And we are now the sole provider of the Abtherix mouse, the humanized mouse that has a full human diversity. And so if you like doing in vivo antibody discovery, we can now provide that service. Everything involved with producing the antibodies, measuring their binding constants, their bioanalytical biophysical properties. It's important to know whether your drug is not only active in biology and biochemistry assays, but actually is up to snuff for manufacturing and stability and the like. And we do all that work. We can create bespoke cell-based assays. And while we don't do animal handling ourselves, we do everything around testing in vivo pharmacology, including help you design the study, coordinating the in vivo study vendor, and we do all the sample analysis and assay development for measuring drug levels, immune responses to drug, or biomarker changes. All right, so that's Mosaic. How do we think about the landscape of intraocular therapeutics? That is, drugs that are delivered intravitreally to more often than not affect retinal diseases, but even potentially some front of the eye diseases. So the biggest advancement in this space was the discovery of VEGF blockers that are delivered intravitreally. The most differentiated originally was a drug called ranibizumab, or Lucentis, by Genentech. And it really just changed the way the world thought about treating the wet AMD, or age-related macular degeneration. And along the lines, and what followed on after that was Eylea, which proved to be even better than ranibizumab. And now VEGF blockade has really come onto its own and created a really a transformative treatment for these diseases, like diabetic macular edema and wet AMD. But people have started to look at new targets. And now, for example, Wnt agonism is something that people are thinking about doing to either add on to VEGF therapy or to replace it in maybe the population. Of course, there's a subpopulation of people who don't respond as well or at all to VEGF blockade. Even though it's the minority, it's still a substantial population. In the case of dry AMD, there's a number of complement inhibitors that are approved, the C5 and C3 blockers, Syfovre and Zimura. There's in clinical development is the C1q blocker, and these are all looking very good, but they're really just delay disease as opposed to really improving vision like the drugs do in the wet AMD. And so new targets are still needed in dry AMD to really affect the disease more than the complement inhibitors, but they're a great first start. So the second thing that people think about doing beyond targeting different biology is to make the existing biologies just last longer so that you don't have to get as many intravitreal injections. The need for frequent intravitreal injections actually limits sometimes the efficacy of these molecules because people just don't want to go into the doctor to have this done. And so if you could create a molecule where you had to administer it less often, we think ultimately the real world efficacy would improve, and essentially through an increase in compliance. There's a number of approaches people are taking to increase or decrease the frequency of injection. And one of those things is to make things bigger. And maybe one of the most interesting approaches originally on that was from the Mosaic Biosciences, or this KS301, which I'll talk about a little more later, which is basically make it bigger, stays in the eye for longer. Hopefully you don't have to inject as frequently. Maybe that's not a perfect solution in that case, as we'll discuss. And then there are people looking at gene therapies, which are longer lasting, and you get the body to produce the therapeutic entity for you. And then finally, there are small molecule inhibitors that are in sustained release formulations like PLGA, classic sustained release formulation in the eye. And those are the sort of approaches that are ongoing now. So let's talk a little bit more about that. So for many years, in fact, the 1950s, It's been known that larger proteins stay in the eye and larger molecules generally stay in the eye longer than smaller ones do. And so the thought is maybe that's a way to leverage decreasing the frequency of administration is to just make things bigger. It's also a reason why even FABs and antibodies themselves, or the original therapies like ranibizumab and Avastin is used offline, and an FC fusion protein, Eylea, have worked so well is that they do stay in the eye for a reasonable length of time, but still the frequency of administration is too high at sort of a month or two or three. But the idea of increasing the hydrodynamic radius is a way to potentially make these proteins even better if we could find a way to do that. And that's where Mosaic has focused their efforts, and I'll tell you more about that in a bit. One standard method people have used for some years to increase the hydrodynamic radius is PEGylation. And that certainly works. Things that are PEGylated stay in the eye for longer than the small molecules or proteins that they're attached to. But PEGylation has its challenges. Two major challenges. One, I think, is perhaps a little more perception than absolute proof, which is that people think that there are toxicities associated with PEGylated proteins, especially in the eye. And there's some publications that summarize some of that work. The second challenge, which is actually the first one on the page here, is that PEGylation of proteins, depending on how you do it and where you place it, can reduce the efficacy, reduce the biological activity of your protein. And so there's some engineering that's necessary with that. And so, we decided, while PEGylation is an interesting possible approach and has been somewhat successful, we thought it might be better to do this a different way. And that's what we focused on. So the great thing about taking this increase in hydrodynamic radius approach that I'll give you a little bit more detail on shortly is a publication, which is a summary out of the folks at Genentech here, which showed that actually the hydrodynamic radius is a really good predictor of half-life. And you can see the plots here. The other really great data that or correlation here that you see is that animal models, particularly rabbit, actually behave analogously to what goes on in human. So if you can show that something works in rabbits or non-human primates, there's a really good chance that that molecule is going to perform better and potentially be an improvement in humans as well. And so, this is perhaps a rare case where the preclinical models are really quite predictive of what goes on in humans, at least with respect to the half-life in the vitreous. So a little bit more about that. Basically, when you inject a drug in the eye, what happens is about 30% of it or 20% of it goes out of the back of the eye through penetrating the retina and ultimately into the choroid and then out into the bloodstream. So everything from the eye eventually ends up in the bloodstream. It's relatively low concentrations because you only can inject so much into an eye. But a substantial part of the protein does exit through the front of the eye, and actually the majority of it exits through the front of the eye and can bathe those tissues. And so you can even think about treating diseases of the front of the eye, like where the trabecular meshwork is problematic. If you could treat that, you might be able to help glaucoma patients where the increase in ocular pressures often caused by trabecular meshwork dysfunction. So it's maybe not just retinal diseases where you can do this. But the big point here to consider is, yes, big hydrodynamic radius does keep the drug in the eye for longer. But, the question is there a level that's too big where, yes, you stay in the vitreous for longer because it clears more slowly out of the back and out of the front of the eye. The diffusion is slower, but maybe you don't get the level of tissue penetration that you need. And one of the problems that I believe happened with KS301, the Kodiak drug, was it was very big. Instead of 100 or 150,000 like an antibody or Eylea, this is now close to 1 million molecular weight. And while it certainly was an active molecule, it was inferior to aflibercept or Eylea. And I believe perhaps part of that problem was even though it stayed in the eye for longer and could be potentially reduce the frequency of dosing, it wasn't penetrating the tissue sufficiently to maintain the efficacy that you needed. So maybe there is a too big that you have to balance. And that's part of what we have come up with at Mosaic, is a technology that allows us to balance staying in the eye for longer, but maintaining tissue penetration. And we're taking a slightly different approach to doing that. There are sort of two ways to do that. One is to just make the protein bigger where the actual molecular mass is large, and that's what Kodiak Biosciences did. So you get an increase of hydrodynamic radius just because your molecule is substantially bigger by covalent bonds of things attached. Or you can increase the hydrodynamic radius by a more PEG-like approach where you actually coordinate water, but you do that with a much smaller entity. In this case, we make a small peptide here called PKE that's fairly floppy, and it adds a modest amount of molecular weight, nowhere near a million, but creates a protein that looks like 500,000 or more molecular weight because it coordinates water and moves more slowly. And that's, we think, a better way to accomplish the task, because we think that's going to help with maintaining tissue penetration. And I'll show you the data that we generated that makes us believe we've solved the problem that way. Okay, so here's the start of it. We did biochemical and biophysical measurements. We started with a aflibercept, or actually we call this the VEGF trap because it's not truly a aflibercept. We don't produce this. This is not produced by Regeneron, which is the company that, of course, makes aflibercept. But we made a biosimilar to aflibercept that we call the VEGF trap, we use that as the starting basis, and so everything's compared to the VEGF trap alone, the analog to Eylea or aflibercept, and then we attached our hydrodynamic radius increasing PKEs. There are three that I show you here: PKE 1, 2, and 3. And you can see the molecular weights on these increase a bit, but not a huge amount. Okay, maybe this one is near a doubling, but this one's maybe only 40% or 15 or 20% increases. But by increasing the molecular weight a little bit with these peptides, you can actually increase the hydrodynamic radius substantially by close to 30 or 40%. And another way to think about that is just to look at how it looks on an SEC column. So essentially, here's the SEC data here. Here's aflibercept or the VEGF trap. And here's what happens when you attach a PKE to it. It looks substantially larger. And in fact, you've gotten near threefold increase in the molecular weight, even though the actual molecular weight that you added is only maybe 20, 30, 40% or 50%. So small increases in molecular weight with the right protein that coordinates water and acts essentially like a PEG can cause a much larger change in the apparent molecular weight, even though we've only added a relatively small amount. So what did that do to biological activity? Well, first we started by looking at the binding constant. And, as I mentioned, PEGylation can cause reductions in binding constant, and PEGylation does increase the hydrodynamic radius by an analogous way to our protein-based PKE that you saw. But in this case, we've positioned the protein and it has physical properties associated with it that don't seem to affect the binding constant for the VEGF trap. And here's the native VEGF trap, and here's the three PKEs. You can see it looks like there's not very much different than in the binding constant in any of these cases. So we've created something that makes the hydrodynamic radius looks much larger, but does not seem to affect the binding interactions. Sometimes you can see effects on the association rate constant. It's rare you see effects on the dissociation rate constant. You see effects on the association rate constant because now the hydrodynamic radius creates a larger molecule, which makes slower diffusion. But as you can see here, we're not seeing that effect of our PKE. We wanted to look at a more relevant biological assay, and that's to actually see if their cell-based assay is affected. And again, here's the VEGF trap, and here are the PKEs. And oops, sorry. You can see there are small shifts, maybe 3, 4, 5-fold shifts in the IC50, but it's not substantial. And so we think we're having minimal effects on the biological activity, either through binding or cell-based assays, by adding these proteins that increase the hydrodynamic radius of the VEGF trap. So we're excited about this. We've basically got a molecule that looks way bigger, even though it's just only a little bit bigger, or a few molecules. Don't seem to have big effects on the activity. The activity is maintained. And so the next question is, how do those do in a rabbit pharmacokinetic study? Here's should say one more thing. Actually, before we move to the rabbit pharmacokinetic study, I will tell you that, of course, as anyone would be with a new approach to a drug, you want to know whether these things affect the stability of your molecule. And as you can see here, all of these molecules are quite stable. We put them in a standard 40-degree stress test. We only went up to 1 mg per mil to start. Of course, one would need to eventually test higher concentrations, but this looks extremely good. There's essentially no effect or degradation happening as a result of these pharmacokinetic enhancers being attached to the VEGF trap. So again, really excited about this. No change in activity. Stability is excellent. Now we can think about going into a pharmacokinetic study. So rabbits are generally a species of choice. And I showed you earlier that the pharmacokinetics in rabbits and any improvements in that are often predictive of what can happen in humans. And so that was the natural choice for the species. We wanted to test each of the 3 PKEs, and we compared it directly to the VEGF trap, the naked VEGF trap. We administered a dose that would be a concentration that's similar to what Eylea is injected into the human eye. We only collected data out to 7 days. At some point, we may want to go out further to get a little bit better data. But the reason why we chose 7 days is that rabbits are often can have an immune response to human drugs that are administered. And so in my experience, once you get out to 2 weeks, you start having potential problems with that. You don't know for sure. So we thought for the first study, let's just look at 7 days, see if there's a strong read there, and then maybe after that, then we can test at longer times to see if we avoid the immune response and can get a little bit more full pharmacokinetic data. We tested not only the vitreous, but we looked at tissue penetration into the retina and the choroid, and also measured the aqueous humor, so we get a sense for what's coming out of the front of the eye as well. So here's the data. You can see the vitreous is over here. The VEGF trap is cleared relatively quickly, between 3 or 4 days. This is consistent with the literature in rabbits on this, so we were able to reproduce that. And you can see at least some of the PKEs are now close to 2 times the half-life. So by increasing the hydrodynamic radius 2.5 to 3-fold, we've now increased the residence time in the eye by nearly 2-fold. And we were very excited about this. But of course, the biggest concern you have is, well, are we still getting the tissue penetration that we need? Because that was our belief on why the Kodiak molecule didn't work as well as it could have, is maybe it was too big. So we thought this is a better mechanism to increase the hydrodynamic radius and would have a better effect on tissue penetration. And sure enough, as you can see, there's actually not a significant difference or substantial difference between any of either the VEGF trap naked or the other molecules that are being tested here. This one looks like it might be slightly high concentration, but this is actually one of the PKEs. So I think these are all roughly equivalent. I mean, we're not even seeing a 2-fold effect here. And so we were obviously excited about this possibility, is that we've now identified something that increases the half-life in the eye, reduces the required frequency of injection based on half-life, but has still got that substantial necessary tissue penetration so that the biological activity in an in vivo system should be maintained. So we did one more thing, which is to see, we've got a new version of potentially aflibercept or Eylea. What would this look like if we modeled how long could this last in humans? And so this is just a kinetic model built on our data. And you can see this is to set up the differential equations are allowed. And you need to account for 2 things here. It's not just how long the molecule stays in the eye. You have to account for the fact that sometimes the target of your protein is produced at sufficient levels that it can essentially functionally clear the drug because it's bound to the drug. So there's no active drug in there. So you want to account for the amount of VEGF that's made in the eye and the production rate and its natural clearance, in addition to how long the drug hangs out and around. And so you need to build all those things into the model. It turns out the biggest effect in this case is how long it stays in the eye, but there is a small contribution from the drug being bound to the target as well, and that clearance mechanism. But what you can see here is we basically used our VEGF trap as a model based on the Eylea data, and said whatever the drug concentration is at 60 days, which is roughly how long you dose in between Eylea administrations, and whatever that drug concentration is, if we can maintain that drug concentration, because we have similar activity of our drug, that ought to be the extension of the dosing frequency. And what you can see is that we've come close, in this case, nearly doubling the frequency. So, we could take now what's given every 2 months, and maybe it's every 4 months, or if it was every 3 months, maybe it's now every 6 months. And so this was an exciting result, and we're looking forward to finding ways that we can partner with people to advance this technology further. Mosaic as a partnered research organization, we want to help other people develop their drugs. We don't develop our own drugs, but we develop technologies that other companies can use to advance their drugs. And so we want to partner to help move this program even further along. So summary of what I told you about. Mosaic is a partner research organization with a fully integrated end-to-end service provider for drug discovery, particularly protein therapeutics. We do work in many therapeutic areas, but probably if we had to pick one where we have the largest amount of business, it is in ophthalmology, and we have a long history in doing that. And because of that, we decided to work on technologies that might enhance ocular therapeutics. And as I mentioned, one of the biggest needs is to decrease the frequency of administration. And we think we now have a technology that allows us to make simple fusion proteins with whatever your active protein is to allow it to stay in the eye for longer. We've seen close to twofold increase, which resulted in a prediction of a twofold increase or twofold decrease in the frequency of administration needed in humans. The approach is to basically increase the hydrodynamic radius of the protein. But we do that not by just making a much, much bigger protein. We make it a slightly bigger protein, but add an entity that coordinates water and creates a much larger hydrodynamic radius without just increasing the covalent nature of the drug. And that maintains the tissue penetration that we think is needed for efficacy. We're actively looking for partners here, and we hope some of you come to us and work with us to help try and enhance some of your proteins. Just a little bit more about Mosaic. We have 80% of our employees have advanced degrees. We're over 50 people now. We're based in the US, and we're an experienced team of drug hunters and sophisticated scientists who know how to make medicines. We look forward to finding ways to work with you. Who are the companies that we work with? Well, we probably work with earlier stage companies more than others. They're the ones that are often trying to do things as much virtually as they possibly can. But we've definitely gone and had partnerships with large pharma and all things in between, and even academic groups looking at very early stage. We're primarily a fee-for-service organization for our research. In the case where you would leverage the technology here, there would probably be something more than fee-for-service in this case if you decide you want to move forward, but we would negotiate something that is mutually acceptable. I think that's the end. Happy to take any questions.
Fantastic. Thank you so much, Eric. Thank you for sharing the scientific rationale and also the practical implications for retinal drug development. So just had looks like 3 questions that have come in. So I'll just dig into those. The first one says, this is really interesting, but it feels that a lot of these long-acting strategies are still evolving. How mature would you say this approach is today, and where are you most focused on improving it?
Yeah, it's a good point, and this is early on, and that's why we want to partner with people to try and advance it. I think the biggest thing we want to do, and my sense in talking to a number of people in the industry, is twofold is good, but threefold is better. And so we would like to see our proteins be a little bit longer acting. The beauty is we're in the process of making further engineering these proteins, making them a little bit bigger, and changing some of the biophysical properties of them, that we think there's a very good chance in the next even few weeks, if not small number of months, that we will have molecules that are actually pushing on threefold and still maintaining the good tissue penetration. I think that's the key where a biggest advancement could be made. We're also looking to see how broadly applicable is this. We've done it on aflibercept. We think it's going to help most proteins, but we would love to partner with people to show the breadth of application, and look forward to trying to do that.
Excellent. All right. There we go. Now we can see your face.
Yeah.
Second one, if someone in the audience is working on a retinal protein or antibody program and is early in discovery, what does collaboration at Mosaic usually look like?
Yeah, so as I mentioned, we want to be your partner in drug discovery. So we're going to basically have a conversation with you about what you're trying to accomplish. We're going to maybe even help you think about what you're trying to accomplish. And we're going to come up with a plan for what we want to do. And then we can just dig in. If it is understanding how this new technology might work, um, you know, we're early on, and so there's a relatively low barrier to entry at this point because we want you to help us prove that this works. And so I think, just to kick the tires, if you will, and test whether the results are interesting, we would be open to just making some analogs of your proteins and, kind of the cost of our work to do that and test them. And then if it's exciting, we can negotiate what the downstream deal and terms are. But just to test it out and see how things are going would be pretty much our standard FTE rates for the time being.
Excellent. Thanks for that, Eric. Last question we have here. How does this strategy actually show up in day-to-day program decisions when teams are deciding whether to push durability further versus protecting activity or developability? How does this framework help guide those trade-offs?
Data. We need to make some molecules. We need to test them. We're happy to work in a close collaboration with you on the strategy for how to design what we want to make and how to test it. We're totally open to making molecules, and you can do part of the testing. We can usually do most or all the testing if you want. And we're open to a hybrid of the two. But, I think getting the empirical data on how well these things work and trying to find the right place to attach them, the NRC terminus, how big they need to be is something we would like to do with you. We're pretty sure we can get close to twofold, and we think it won't be long before we can show we can do even bigger. But to the extent that you can help us show the breadth of applicability and maybe enhance some of these things, we would like to partner with you to do that.
All right, that's all we had for questions today. Eric, thank you so much again for this great presentation. Thank you for taking time to answer those questions. And thank you everyone who submitted, everyone who joined. Just a note, a recording of this webinar will be going out to all registrants, and we'll be following up with you in the next day or so. But in the meantime, if you have any questions for Eric or for Mosaic, you want to have a chat, we'd love to talk with you. Please reach out to sales@mosaicbio.com. And thanks again for joining us. We'll see you at a future webinar. Have a great rest of your day.